Welcome to the 2024 Vermont Organics Recycling Summit, organized by the Composting Association of Vermont in partnership with the Vermont Agency of Natural Resources. I'm Natasha Duarte, the director of the Composting Association of Vermont. This year's summit is being held as a kickoff event for International Compost Awareness Week, which starts this coming Sunday, May 5th. The International Compost Awareness Week is the largest and most comprehensive educational initiative of the compost industry. So it's a lot of fun to organize our Vermont Summit in partnership with this group. The theme this year is Compost, Nature's Climate Champion. And before we get going into this session, I just want to give a shout out to this poster here on the right. This is from a project that UVM students in Dr. Deb Nair's Composting Ecology and Management class do every year. The full collection of the student International Compost Awareness Week posters is available at compostingvermont.org/VORS. And if you click on those images in the gallery on our website, you can actually read the students' inspiration for the creations and how they connect with this theme. So we've been really enjoying and appreciating that collaboration with the University of Vermont. I'd also like to take a minute to thank all of our sponsors and exhibitors. These include Community Bank, Eco Products, Addison County Solid Waste Management District, AgriLab Technologies, Casella, Food Cycler, Nature Cycle, Vermont Natural Ag Products, Viably, CV Compost, the Vermont Produce Program of the Agency of Agriculture, Food and Markets, and Woods End Laboratories. And it's really with their continued support year after year that we are able to put on a full week's program free, with the whole virtual program being free of charge to all of you. So huge thanks to them for their continued support and letting us bring you today's program. We're in the fourth session, One Man's Journey to Soil, and we're going to be hearing from Rob Kurth, who is a retired engineer who's been gardening long enough to know that the secret is in the soil. Robert loves to talk about the symbiosis of plant and soil and to explain as much as we know today about the largely unseen work of soil biology.
All right, well, thank you so much for the kind introduction. And, I've kept my eyes on this seminar over the years. But about a year ago, I started thinking about where I've been as a gardener and where I've been as a person. And I wrote a piece for The Natural Farmer, which is the quarterly publication of the Northeast Organic Farmers Association. And I entitled it One Man's Journey to Soil. And it was meant, I think it was delivered as a humorous piece. Frankly, I find lots of parts of my life have been humorous and absurd. And one of the things that kept me grounded my whole life has been gardening. And I just want to take a minute to acknowledge somebody who was a huge influence in my gardening, and that was Evelyn Carter on Wilson Street in Burlington, Vermont. And, I was about 10 years old and wanted to play wiffle ball in the backyard, and she let us. But she had a vegetable garden back there, and if you hit the ball in the vegetable garden, it was an out. So you didn't want to pull the ball down the left field line because you were going to get an out in the garden, unless you hit it all the way into the Sheehy's yard, in which case it was a home run. But I remember fetching that ball from that garden, and my parents did not garden. My father seems to have had a pesticide thumb on anything he touched, but Evelyn didn't. And so sneaking in there and sneaking a carrot occasionally and a green bean, etc., somehow that all settled in me. And so about now 50 years later, I'm going to tell you the story a little bit of how I got here and what I think about gardening today. And what I think about compost. That's going to be in there too. So you can go ahead to the next slide, please. So for a seminar, I thought maybe I should rely on some science, and that informs my story, and I hope to inform you about the science. And there are really three things from a sciencey vantage point that I want to talk about. The first is the National Resource Conservation Service Soil Health Principles up in the upper left here, Soil principle 1 of 4, soil armor. We'll talk about this later, and if all of the audio works well, we'll hear a couple of clips from some experts in that area. The next area is the soil food web, and this for me marks the change in science that's taken place in about the last 40 years. And Dr. Elaine Ingham, among others, but chief among others, has said about her graduate work as a student at Oregon State, I believe it was Oregon State, looking at the biology in soils. And what has resulted is national and international awareness of the importance of microbiology in our soils for many reasons. And I'm going to only talk about the gardening reasons to do that, but there are wonderful environmental reasons and perhaps even climactic reasons why we all need to understand life in the soil better. And Dr. Elaine Ingham has set that out for us, although there is very much new science. She paved the way, showing how important the soil food web is to a healthy functioning soil. And the last thing I'm going to talk about, I think, is photosynthesis. And photosynthesis is underappreciated, believe that or not. I believe it, that soil is the product of plants gathering energy from the sun for however long that's been going on. And it's really it is the only source of energy that any of us has, is from the sun, mediated through plants into the soil. And the symbiosis between plant life above the soil and plant life below the soil and the interactions with the soil, this symbiosis is just so critical to our understanding of our planetary health and our plant health. And I'll cite a few people who've given this some thought. Next slide. So I want to start off with these NRCS soil health principles. They've been around for a while now. Their number has varied over the years. And so today we're going to talk about 4 soil health principles. The first one being soil armor. Always keep the soil covered. Nature certainly does. We have no dispute with that. Nature will always cover the soil. Minimize soil disturbance. There's some easy things like tillage. There's some hard things like fertilization. But anything we do to the soil, we have to understand we're disturbing it, and we should minimize that disturbance. Continual plants and roots. So green growing plants using photosynthesis is what is the energy driver for the entire system. And the longer you can have green growing plants and the more diversity of green growing plants you have, the more energy we can put into this system. And as it cycles and cycles and cycles, we end up with healthier and healthier plants. Mother Nature certainly does without adding anything. And then lastly is plant diversity. I mentioned that. This is such an important topic. I'm not going to cover a lot about it, except, well, I'll have some comments, but they may well be sociological comments more than anything on plants. Now, number 5, livestock integration. When the USDA and the NRCS first came up with this, they were working with researchers in North Dakota. And in North Dakota, the livestock is not just integrated, it is the whole deal, is livestock up there. And so the USDA worked out these things, and livestock was in there, that you needed the bacteria on the tongue of the cow in your soil, which is true. If you can have a cow licking your plants, it'll help. But we can't do that, well, not all of us, and certainly at garden scale. I mean, there's some big backyard gardens that have some goats and pigs and chickens and all of that, God bless them, but not at a community garden and not for most people. And so they added a 6th principle, which was context. We have to look at what's going on in context. And I'm going to talk about my context. But once they started talking about context, they actually fell back and said, okay, it's just the first 4 principles. And so most talks you hear today, they will only talk about those four. Livestock was impossible, and then context was only there because of the livestock. But in fact, we all have a context, and I have mine. So let's get started with that. I think it's the next slide here. Nope. Okay. Well, it is. It's a different context. It's the context of the community garden where I do my gardening. This is in Essex Junction, in the village of Essex, well, the city of Essex Junction. Now on West Street, it is a 90-plot community garden. It's surely one of the largest in Vermont. We've got an aerial picture of for you here. And in the top left are roughly 60 plots in one large field. And over towards the right and then the middle is a smaller plot, and that's got 32 plots in it. We are on Sand Hill, and the context of this soil is beach sand that's been tilled for 40 years. And there's no life in this soil. I was first at this community garden from 2011 to 2015. My wife and I were living in a condominium, and the last thing you were simply not allowed to even own a shovel, as far as I know. So we went up to the community garden, and I gardened there for 5 years. I had 2 plots, 1,250 square feet. I never saw a worm, not one, in those 5 years. And my wife and I managed to buy a small piece of property, big enough to garden on, and so I left the community garden. This is the way people garden here, and they do it successfully. And therein lies perhaps the challenge, some of the challenge of getting this new science across to people, is that we have different definitions for what we mean by success. There's an enormous tension in the American community garden that I've experienced, and I was gratified to find that the sociological researchers of the world have actually paid attention to sociology in the community garden around the world. And one of the biggest tensions is here in America. The tension is between the laid out grid with least services versus a more collaborative style of gardening. Anything from lots of people working together because we make noise, perhaps, to just simply gardening differently. No-till was a hard sell. It took 4 years to get no-till into this smaller field, but we're there now. And the people who are in the no-till area want to be there. But it's pro-grow land, 5-3-4 organic fertilizer. You put that on that sand and you will grow vegetables and you will grow weeds. And if you are good at managing weeds, then you will have some terrific vegetables out of this soil. You will also add to what is perhaps 50 times the amount of phosphorus that we would like to see in this soil. This land has been agricultural land for 100 years or more. And there has been so much phosphorus poured on these fields. Now, phosphorus runoff is surely a problem, but phosphorus accumulation is also a problem, and that's what we've got. Once phosphorus gets down in the soil, it doesn't move anymore. It stays put and it starts reacting with things and tying up some of your others. So calcium carbonate, no, I'm wrong about that. Forget it. Forget I mentioned it. Phosphorus will tie up other critical nutrients. And that's what we've got here. And if you want to continue doing this, and if you want to continue putting ProGrow 5-3-4 or 2 inches of compost, you can grow some fine vegetables here. Then that's the way you'll have to do it, unless there's another way to do it, and that's what I'm here to talk about. So go ahead. Next slide, please. Okay, so a little more about West Street. The USDA soil series is called Adams. The Adams series consists of a very deep, excessively and somewhat drained soils formed in glacial fluvial, we're beach sand in Essex Junction. And those of you who live on Sand Hill, you know what that stuff is. It's beach sand, and the only reason that it's not actually beach sand is because something is growing on it. And if you don't grow anything on it, you have beach sand. So there's a long history of agriculture here. Before this was a community garden, it was a tree farm. Before it was a tree farm, it was a cornfield. Before it was a cornfield, it was something else. But this has always been agricultural land. It's fertile soil with too much of everything except soil life. And that is directly related to the management of soil. In managing a garden soil as though we are practicing agriculture efficiently. And so most of these soils for the longest time were tilled not once but twice a year, once in the spring, once in the fall, for about 30 years. After that, the volunteer who did that, he was getting older, he only did it once a year. And then the volunteer got so old that he couldn't do it anymore. And it was at that point they allowed one of the fields to go to no-till. So in 2019, we transitioned that smaller field to no-till or low-till. The large field is still tilled once a year. We did offer a service. We said this is a no-till field, but if you insist, then we will provide the tiller. You can till it yourself. And that's the last thing we did last year. I don't think there are any plans to do that this year. It's a no-till field, and if somebody needs it tilled, I don't know, they can raise their hand, they can ask for help, I suppose. So, and help them, we will. I'm not here to argue with the way anybody wants to garden. So anyway, that's the context of West Street. I think the next slide will get into the context of me. So go ahead and advance it. Nope, more on soil test. Here's the soil test, Wednesday Sue. We got a lot of everything except trace minerals, especially boron and sulfur. Those leach out of those soils. You can go through this, and I certainly did go through this. I took a class up at UVM, soil science, plant science 162, which is the introductory soils course. And so I've been through these things a few times. This soil is really pretty good. If you have some trace minerals, add them. But other than that, it's really pretty good. So go to the next slide. Okay, so that was West Street. Now here's my context. I told you about Evelyn Carter's backyard playing wiffle ball. When I first started gardening at about the age 22, maybe, I became a J.I. Rodale fanatic, organic gardening magazine. I was living in Virginia at the time, and I was, as instructed, I was double digging the clay. And what I really did was form a bathtub. So I loosened that soil so deep and allowed so much water to get in that bathtub that everything I planted died. Now, thankfully, all I had planted were 6 tomato plants, but it was a pretty quick death. And so that was my first experience of gardening. So, following the precepts of organic gardening, I set out to build the Cadillac of composters. This was made popular by Bob Crockett, I think his name was, on the Victory Garden back on PBS in the 70s. And it's a big affair. It's 12 or 13 feet long with 3 bins, and build it out of whatever materials you want. Mostly, I built it out of pressure treated at the time, because in 1981, that seemed like, 1983 maybe, that seemed like a good thing to do. It would last a long time. My employer had other ideas, and I was transferred from Virginia to New York State, and they were unwilling to pay for the transportation of my Cadillac composter. So I actually built it and never used it. But I found myself in Pleasant Valley, New York, and my wife had an interest in small livestock. So my kids and my wife and everybody had some stuff out there. We had chickens and we had pigs and we had goats and we had all of that stuff. And I had raised beds, and I double dug the raised beds, and I had all of this compost. And the problem with that was I've got so much compost, I don't know what to do with it. Personal life interfered, and I didn't remain at that garden for that long anyway. And then I moved to heaven, and that was in a town called Millbrook, New York. And this was an 80-year-old clay loam with chickens that was the most gorgeous soil that I've ever seen. It was at least 2 to 3 feet deep. And it was that way because it was on the wrong side of the tracks in 1920. And in 1920, and if you moved to Millbrook, New York, and you were Italian immigrant, you were living over there. And that's where they lived. And the soil was gorgeous. This place could grow anything. I mean anything. Now, the climate in the Hudson Valley is awfully good. So there's that. But this soil was magnificent. So then I fell in love and moved to Vermont and started over again. And this time I was studying The Agriculture, which was by Rudolf Steiner, written in, I believe it was 1923, 101 years now. Steiner is the founder of the Waldorf schools. He has something he calls anthropophagy, which is his take on the universe, more or less. And in the garden, he had some very particular ideas. In the 1923, he was lecturing, and all of the lectures was compiled into a book called The Agriculture, and he basically told the farmers in his valley in Switzerland that they were not to use these new chemical fertilizers. And he gave them some different approaches. Those approaches are very, very different. The man who thought of anthropophagy and the Waldorf school and paid so much attention to so much in his life said the thing you have to do is take animal manure, you take cow manure, and you put it in the horn of the cow that you slaughtered, and you bury that 1 foot under the soil surface in the fall. You dig it up in the spring. You take the contents out that is now thoroughly rotted and filled with biology, and you put it in a 5-gallon bucket of water, and you stir the water manually for an hour. And you stir it one direction, and then you reverse course suddenly, and it's the breakdown of the vortex that allows the forces of the outer planets to enter. So then I got sober, and I started gardening a different way. Now, I don't mean that to be funny. That's what happened. So I'm a member of a 12-step program, and at this point my life changed in many ways. And they told me I had to find a power greater than myself. And that was easy, because that was in the soil, as far as I was concerned. There was a power much greater than myself. And that sufficed for me. And I was privileged to speak from the podium at these 12-step meetings. And I remember distinctly back in 2009 saying, out at the very furthest tip of the finest root hair of the plant, science doesn't know what's going on. It's just this soup of animal, vegetable, and mineral. Well, based on science, I've had to revise what I say at the podium, because we do know what's going on out there in broad stroke, although there's so many details still to figure out. But we do know what's going on out at the tip of the root hair. And by golly, it's nothing we thought that was happening out at the tip of the root hair. Now, for me, it's still a miracle. It's still a power greater than myself, even though we know more about it. Because it really is truly a miracle what's going on out there. So things changed for me. I went to the master gardening class in 2015. I retired, and I had a little bit of money to take a class, some kind of retraining. And so I took, Plant Science 162 at UVM with Dr. Joseph Gorez. And I became hooked on this chemical idea of how soil functions. And so in that class, we learned that ionic minerals move not only in response to electromagnetic forces, but they also diffuse where there's a concentration gradient. And so the idea is that the plant is removing nutrients from close to its root to be incorporated up into the plant, And we've now created a concentration gradient so that more calcium will reach the plant root and they will move like that. That's a good theory. It hasn't been disproven. It's probably, I think it's true, because we know that diffusion occurs everywhere. Where there's a concentration gradient, there is diffusion. We know this. And there's no questioning it. But it's not the whole story. It's a good part of the story, but it's not the whole story. And that's what I've learned in the last few years. And that's been a concentration on regenerative methods using natural resource conservation service practices that are all focused on biology. Not so much, although there's not a farmer there who won't test their soil, because if you're short of something, you better supply it. At least, that group grows, but maybe we've got 16 or 17 elements at the moment that we consider critical for plant growth. And if you are missing them, you should find out if you can add them. But other than that, especially at garden scale, we need to turn our attention to biology. So next slide. And that's what I've done. So there's my context. So go ahead. So remember, I told you that there was this ideal soil. Well, I didn't mention that. As part of PS 162, you get into this soil testing and interpretation of results, etc. And I spent some time on that. And so this is from 2020 data. This is the ideal soil recommendation. There's actually a book called Ideal Soil 2.0. And the concepts in there can be reduced to a spreadsheet, as was done here. And if you could read the fine print over on the right, I got to put on my glasses just to remind myself. So if I need a little magnesium, I'm going to add some magnesium sulfate at the rate of 39.2 grams per air. Air being roughly 1,000 square feet. I need some ammonium, because I'm not against adding nitrogen to the soil, especially if I've got some on hand over there. Copper, a little zinc. And if you'll advance to the next slide, please. This is a waste of time. It was for me. It was a complete waste of time, because I was completely overrun with weeds. I am overrun, as most gardeners are, with cutworms. Whatever level of pest you might think, we've got it. I've heard it described that the gardening in a community garden is like graduate school, because you have every pest and you have every disease known to man is right next to you all the time. And so the idea that I'm going to fundamentally change the character of the soil enough through chemical additions became ludicrous to me, because they showed no results at all. And if you look back at the soil test, you say, well, duh, you weren't actually missing anything. You could argue about whether you're deficient in something or another, but it's not actually helping you get better results. So I was led on to something else. So next slide. Okay, why is Jay Fuhrer's beautiful face not here? Oh, there he is. So I want to play a short clip by Jay Fuhrer. I think it's a few minutes, but it's not very long. But Jay is going to talk us. He was the leader of that NRCS soil health team up in North Dakota that did the work, the original research for USDA on this, and he's a great spokesman for it. So if you'd play that, please.
All right, I'm going to go ahead and try to, whoa, sorry, people. Oh, is it a link to an external video? Do you know?
It is. Do you not see that?
Here we go. Hang on one second. And I'll make this big and just let me know if you can hear it. Otherwise, I can stop sharing and tweak the settings.
Okay.
Can you hear that? No. Okay.
Muted.
Hang on one second. All right, let's try that again.
Well, I think what we want to concentrate on in this talk
Can you hear
is
that now?
basically soil regeneration. Because my goal is to farm forever. I'm not interested in a short-term plan. I'm interested in something that allows us to maintain a high quality of food for forever, for generations to come. And I think in order to do that, we have to take a look at the past and see where our weak points were. Certainly carbon levels are probably one of the more critical factors. And so we've concentrated initially on bringing soil health principles to stabilize the soil and then bring the carbon levels higher. And then we would see some subsequent impacts from that, like increases in soil biology and increases in resiliency. And it's really a lot of little things that we bring together that start to make these changes and these improvements. And so it's been kind of a road of information and education, and certainly ones of working one-on-one off the back of an in-gate on a pickup at the edge of the field. And I think the boots on the ground is, to me, what makes a change one field at a time. The first principle would be armor, or putting residue, either a green plant or dead litter residue on the soil surface. And so that, to me, stabilizes the soil surface. Takes care of a lot of our rain impacts, takes care of a lot of the evaporation issues, temperature, all of these things start to moderate. So the first thing I like to do is armor up a field, put residue or a green plant on it, something that covers it. Second one is we started to reduce our disturbance. And so we started to minimize disturbance. And it initially started fairly straightforward. If you were tilling that field 3 times a year, how do you get to 2? If it was 2, how did you get to 1? And eventually we took all the disturbance out. And so you start moving down that road, putting the armor on and then taking the disturbance out. The third principle was we increased our diversity. And so there's 4 major crop types: cool season grass, warm season grass, cool season broadleaf, warm season broadleaves, and we try to bring all of those crop types in. And as we increased diversity on the soil surface, our biology increased. And so these two go together and they're direct reflections of each other. The next principle that we put together was a continual live plant. And that's when we brought in the cover crops. And so as a cash crop would be harvested, we'd start to basically chase the combine out of the field with the tractor and drill and put in a new seeding. And the cover crop was an opportunity to give us what we didn't have. And so if we were missing one of the major crop types, we could pick that up in the cover crop. And then also with the cover crops, we brought in multi-species cover crops, and that allowed us to adjust the CN ratios. And so the carbon-nitrogen ratio became a real critical factor because now we could do some adjustment of it.
Okay, stop there.
Whereas if you have a monoculture,
Great. My apologies when I was rehearsing, well, not rehearsing, but when I was looking at this on my computer, they're all embedded and they've got a start and stop time attached to them and all of that, but that's okay. I think we might listen to one other of these and not much more, but I'm certainly willing now to pause and ask questions. If anybody has questions about those 4 NRCS soil health principles, I'd be glad to try to address them. I don't intend to go over them anymore otherwise. Any questions?
Okay, great. We haven't seen any come in through chat yet, but folks, I'm going to suggest that, Rob, you keep going, but as you have questions or if you want to add a comment, go ahead and put that in chat and I am monitoring that.
Yes, please. I'd love to see your comments. You're hearing mine, so I'd love to see yours. So we can advance to the next slide. That'll be fine. And we're going to change gears here, and we're going to go to that second broad topic, and that is the soil food web with Dr. Elaine Ingham. This chart is everywhere now because it demonstrates a fundamental concept in biology of a trophic level. And so what you see here are roughly 4 trophic levels. Those are the things that eat each other. So starting over on the left, around the root zone, we should, in a good healthy soil, we should find a lot of bacterial and fungal life in that soil and even extending into the plant itself. But it should be an area that's rich in biology. And that biology happens because of these trophic levels. The richness of the biology happens that way. Because nematodes come along, and some nematodes are going to nibble on a root, and some nematodes are going to eat bacteria, and some of them are fungal feeders, and you have nematodes that eat other nematodes, and that's the next trophic level. And then something else comes in and eats those, and something else comes in. And one of the important points to understand here is that each point in that process, from trophic level to trophic level, the actual nutrient requirements of the next level go down. And so a nematode eating a bacteria actually doesn't need everything that's in the bacteria, and it excretes it. And you'll find that at each stage, there are decomposition products being made because there's too much nutrient for the next trophic level. And look at any trophic level you'd like. Look at any animal you'd like. If they're eating and they're healthy, they're going to excrete something that they don't need. And so it's those products that are now become more available to the plant, exactly where the plant needs them. Now, this is what Elaine Ingham worked out. It seems like something that they probably worked out in 1870 as well. And by 1890, I think they understood this very well. But we had to reinvent this stuff. And Elaine Ingham was one of the pioneers in doing that about 40 years ago. So I want to talk some more about biology. So if you please advance to the next slide. And these slides are from Elaine Ingham. Go ahead and advance the slide, please. So this is directly from Elaine Ingham. You know, what is it about this biology? Why is this important to us? And she summarizes it in this way. First is to suppress disease, competition. So no one microbe or fungus gets the upper hand on anybody else. And so the rhizoctonia that might be lying in wait for your plant to kill is kept in check, and it actually performs soil health functions as long as its numbers don't ever get too big. This idea of one microbe or life form inhibiting the behavior of another life form is critical, and they keep consuming each other. They just keep consuming each other. This whole thing adds another layer, huge layer, to the nutrient pool that's available to the plant. If the fertilizer we apply gets incorporated into microbial bodies, then it doesn't leach. It ultimately becomes part of the humic substance of the soil. Of course, there are ways that could be disrupted, even by nature. But if left alone, we will have an accumulation of nutrients in a healthy soil that will be retained. The nutrients are available at a rate the plants require. In fact, in a healthy soil, the nutrients are available at a rate that the plant requests. We know more about that today too, and I'll talk about it in a little while. Soil life is so valuable to decompose toxins, and there are plenty of toxins in every soil everywhere, and let's hope they keep doing that job for us. And then lastly, and I think Elaine Ingham would say most importantly, these microbes are actually critical to building soil structure. So the soil aggregates and micro aggregates inside the aggregates, all of that is held together by a biological glue. It's called, one of the major ones is called glomalin. We didn't know glomalin existed until about 1998. But that's the glue, and that glue is made by the microbiology to hold that soil together. Really remarkable. We'll see some more of that later. So I've got a couple more slides from Dr. Elaine on biology. Go to the next one. Here's her illustration of the nutrient pools in soil. And so I was talking about the availability of calcium by diffusion across a gradient. And that's true, that is available nutrients, but in the total pool of nutrients, it's a very small amount. She represents it by that small gray box at the bottom. Soluble fertilizers moving in response to concentration gradients. And then she points out the large box that says if you take the nutrients that are contained in all of the biological beings in the soil, they're much bigger than what the soil alone can store as just a mechanical growing medium. Okay, next slide, please. Next slide. There we go.
Yep.
Okay, so Dr. Elaine also pioneered the use of microscopy. When she did her studies of soil microorganisms, she did it by counting them in a microscope. And so there's some, I think I see flagella on whatever this is, so it's some kind of protist. So it's one of the higher orders, probably the second tropic level are these protists. And they're mean, and they eat. That's all they do is go around and eat and multiply. But somewhere along the line, they're doing us a service. And she was able to define that in published peer-reviewed research of the importance of looking at soil biology. And then we'll just go on to one more slide from Dr. Elaine, and this one, the next slide is a fungal portion. So one of the central points that Elaine Ingham made was that by disturbing soil, we are interrupting the fungal components of our soils and constantly setting them back to be merely bacterial in nature. And she did the studies to show that the fungal concentration in a mature forest is exceptionally high, and then the fungal element in beach sand is near zero. And if you know that going in, then you know something about what crop might do well in that soil. And so she's studied this, I mean, from zero life to full mature forest. And she finds, not just she, science finds that the nature of the soil biological activity is going to change and mature as the soil changes. Okay, next. Okay, so how do I focus on these things? What can I do to improve the situation from a biological standpoint? And so we work a lot with soil and seed inoculants, and I use a lot of compost, make no mistake. But I also use it as a simple biological inoculant. You know, a half inch of compost may be all that I need to assure myself that I'm doing what I can to add biology. This bed, for example, I don't want to disturb it. I can't pile a bunch of compost on the top. I am not a convert to the raised bed method of putting down a bunch of cardboard and then a compost on top of it. In my experience, that dries out so fast that I can't garden in it. But I like compost. I use it as an inoculant mostly, and not a direct soil amendment. There is simply not enough compost that I can possibly add to this sand to change the fact that this is Adams series sand, 30 feet. That's what it is, and it's not going to change. What I can do is add some biology to start to glue that soil together, millimeter by millimeter by millimeter. And it can be done pretty quickly. So I do apply bacterial and fungal applications. There are many, many commercial sources, and you know, there you go. There are many commercial sources for these bacterial and fungal amendments. Or you can make your own, which is pretty fun. You know, fermenting nettle, for example. Nettle has incredibly high concentrations of many trace elements. And by fermenting it, it gets complexed into bacterial bodies that can then simply be sprayed on the field. That's the theory. That's what I'm trying to do. Bacterial and fungal nutrition. So I add a lot of molasses. It doesn't take much. A lot of molasses for the soil is not really a lot of molasses compared to a jug of it. But molasses, milk, various anaerobic brews, anything I can do to encourage biology in the soil. Trace mineral nutrition, I do some of that too on this sandy soil. We've got boron, sulfur, magnesium, manganese, copper. They all tend to leach out of the soil. They're at relatively low levels in our soil. So I do add those as well. But those minerals have to pass through biology before they can become plant available in those trace amounts. Moisture management becomes very important. Once you introduce all these microorganisms, you have a vested interest in keeping them alive and active. And so moisture management becomes very important. And then inspect and measure, pull up a plant, hopefully a weed perhaps, but pull up a plant and look at it. Look at the roots. See what's going on in your soil. Do worm counts. I told you I gardened for 5 years on a double plot without seeing a worm. Well, 2 years ago, I had one spadeful of dirt that had 23 worms in it. So you can test yourself on this. If what you're after is to encourage soil biology, and if you believe that this soil food web is true, then if I have an abundance of worms, it follows that I have the rest of the food chain there too. So worm counts are a very, very simple proxy for, is this soil healthy? I don't want to talk about jumping worms today. But anyway, and then you can inspect and measure yourself in a microscope. I have a microscope. They're not that expensive. But if you wanted to go about and make a bacterial brew in your basement, you can actually check it before you apply it to the field. You can put it under the microscope and say, did it work? Did I, what are those things? Are they protists? Are they bacteria? Are they fungi? What was I after? And did I achieve it? So next slide, please. Now I want to talk a little bit about advances in soil biology research. This is very interesting. It's to me. It is probably unnecessary as well if you believe again in that soil food web. You don't really need to know any more than that. And yet, since that soil food web was proposed, there's been a lot of science that's gone on since then in the last 40 years, and it is filling in the information about why the biology is so important. And so we have whole new areas of research that are going on, and I'm going to take you through a few of them. I'll just talk about them. I'll give you some names of who you can follow up. If you want to find out more about this, there'll be some references at the end too, but one of them is biopriming, it's called. I call it seed inoculation. We've all been doing this for a long time. In the case of legume crops, we've been inoculating them with rhizobia bacteria because they may not be present in your soil. So it's easy to buy a little bag of it and add it. Well, there's a lot of things you could add to your seeds. Nicole Masters has one of the simplest ways, and that is for you to maintain your own soil biological brew in perhaps a 1-gallon bucket. Take some healthy soil, take a little bit of molasses and a little bit of milk and stir it up. And when you're ready to use it, add some clean water. Soak your seeds in that for a few hours. Then let them dry out. It doesn't matter. The microbes simply go dormant. They don't die, at least not quickly. But they're all on that seed coat. So you can inoculate them with just the most diverse biology you can think of. All right? Get soil samples from different healthy gardens. If you see a healthy plant, no, I was going to say rip it out of the ground. But if you happen to be inspecting a plant because you wanted to look at its roots, and it turns out that those roots look really healthy, then chop them up and add them to your gallon. And they'll be there. They'll be there.
Rob,
The next
just a quick time check. You have about 10 minutes left.
10 minutes. The plant health pyramid, photosynthetic efficiency. This is by a man named John Kempf. I've got more on that later, so I'm going to skip over it here. In fact, I've got a little more on all of these, so I'll just go over them real quickly. Quorum sensing is an area of biology that is studied in many different biological disciplines, put it that way. And we'll go through that and how that applies to microbes in the soil as well as the human gut. Rhizophagy, this is the biggest one that's new. It turns out that plants are farmers, that they grow bacteria near their root systems in order to consume them. So the plant nutritional model that relies on diffusion of ions toward the root does not account for the fact that there are bacteria all through the entire plant, and they've been consumed by the plant in a process called rhizophagy, or root eating. It is not us eating roots, it is the root eating biology. And that is credited here in the United States anyway to Dr. James White, and we'll see some of that. And then the last one that I'm just going to mention is genetic expression and epigenetics. There is a lot of function in even the simplest microbes in the soil. And depending on genetic expression of the soil and of the plants, we can have very, very different outcomes. So let's try to finish up quickly. You go ahead on through. Next slide, please. Oh, Nicole Masters, I told you what she's doing. This is a wonderful little video made for the city of Los Angeles to encourage its residents to be better gardeners. She talks for about 5 minutes about how to make that microbacterial brew and how to add it to your seeds before planting. Simple as that. Okay, next one. Okay, the plant health pyramid. This is John Kempf. John is a polymath. If you take even a few minutes to listen to John talk, you will understand that quickly. He distinguishes between what the plant can do on its own in terms of its own health and immunity, and what is dependent on bacteria. And he draws these 4 different levels. The first one is concerned with photosynthesis, and John Kempf spends a lot of time and makes a lot of money applying precision doses of fertilizers in foliar spray form. And it's his assertion that most plants are photosynthesizing at only about 20% of their inherent capacity, and he thinks he can get his up to about 60%. At that point, you start to have complete protein synthesis. So now you have a plant that's strong enough and healthy enough to actually assemble proteins, not just amino acids. It goes on, and this is the distinguishing layer, he says, between passive immunity and active immunity, is active immunity cannot be achieved without the help of the soil. It must be in symbiotic relationship with the soil to achieve those higher levels of complex synthesized molecules, be they lipids or phytonutrients. So anyway, worth a peek for you want to know about the importance of biology to plant health. Next slide, please. In fact, you can skip 2. Okay. That's the details. Quorum sensing. Quorum sensing is a phenomenon where bacteria respond to signals within each other. There's a very simple model that we're all probably familiar with. It's just the common cold and flu. You know, those microbes, those viruses are with us all the time, but they don't become virulent until they achieve a concentration that they all agree, okay, now all of us get nasty same time. We know this is the case, and it's called quorum sensing, and it happens in the soil too. So you can have all of those disease organisms in your soil, but if they're being outcompeted, they never achieve that quorum sensing level of says, okay, let's destroy this plant now. There's never enough of them around to do that. So next slide. That's probably a video we can skip. This is an excellent video by a woman I love. And I could probably
We
even.
can provide links to the videos. So for
I-
folks who want to check them out with the recording, I'll make sure
Yes.
that active links are provided.
Right, I put them in a bibliography at the end of
Oh,
this.
great.
But this is Christine Jones. Okay, so rhizophagy. This is root eating. Let's go to the next slide. We'll try to get this done quickly. Okay, rhizophagy. So rhizophagy is root eating. What they're eating is bacteria. And if you follow this diagram, we'll start over, I think it's on the right, at the meristem of the root. That's the growing end of the root. Has some very, very special properties out at the end of that root. And one of them is that it can swallow bacteria. They don't know the mechanics of how it's able to do that, but we absolutely know that they eat bacteria. They hit them with a superoxide, which strips away their cell walls. And what's left is this protoplasmic stuff that the plant can consume, except the plant doesn't consume all of it. It leaves some of the DNA information around. And in a miracle, that DNA gets re-injected out into the soil through a root hair. In classical agronomy or classical plant science, a root hair is absorbing nutrients, but it's at least a two-way street, if not a one-way street the other way. And in a healthy plant, it is actually ejecting bacterial DNA along with a food source for those bacteria to reform themselves and to go out and do it all again and to go forth and multiply and come back and get sucked in by that plant again. And nobody knew any of this was true even 10 years ago. So I'm here to tell you, the soil science is focused on biology. And the way to achieve biology, at least for the average Joe, is listen to the NRCS soil health principles and practice them. They're not hard. I was absolutely amazed the other day. I've been out in the garden many times, but I realized that at the moment I don't have any work to do. All of my soil is covered, either with a green growing plant or with some straw, or with plant residues from a crop that I'd planted the year before. And it just keeps getting easier every year. Now, it's really hard to subject that to any test that we might imagine. If you want to measure crop yields, last year was a disaster for most of us. Pest pressures. Weed pressures last year were awful. You take a nice fertile soil with lots of pro-grow in it and add water, and we get weeds. Last year was a disaster. But I'm telling you, I've been out there this year and that soil is ready. And it's no work. And I encourage all of you to try it. So that's what I've got.
Great. Thank you so much. We really appreciate your participation and presentation, Rob. I'm going to go ahead and stop sharing my screen here. We are right at the hour. I want to be respectful of people's time, but if anyone does want to have any comment or quick question, we can certainly manage that. That was really interesting information and a track through your context, Rob, and a great overview and summary of a lot of the different principles that we put to work. And with that, I'm just going to say again, thank you so much, Rob, for joining us this year. And thanks to all of our participants. And we hope to see you all back tomorrow for some of our sessions.
Thank you.
Thanks so much, everybody.